Good morning, boys and the girl. Today, as part of our live classes, we are going to discuss about the same chapter, cell, the unit of life. And in that, in our previous sessions, we talked about plasma membrane, anatomy and physiology. And in that, we talked about most accepted model called fluid mosaic model, proposed by two scientists, Singer and Nicholson. And that is the reason why I said you practice once. That is sometimes a 3-mark question will come. And second, I told you in the beginning of this chapter that this chapter is not only important as far as your CBSE board exam is concerned, and it is very, very useful for day-to-day life also. You should know what you have inside and what are the functions that these cell organelles are performing. Then only you will have an idea how your body is functioning. This is utmost important. And the next one is, it is very important as far as your UPSC NDA is concerned. As all of you know, coming April, you will be taking your first attempt in your life, UPSC NDA. Lot many questions will come from this chapter. All cell organelles, at least two questions from this chapter, means 8 marks. And you know, Pallavi, 8 marks in UPSC is very big margin. With 1 mark, you will have at least 5,000 to 10,000 ranks difference. Imagine 8 marks. So focus on this chapter, right? We go next, cell organelle, second one, cell wall. As all of you know, cell wall is the toughest one. And it is tough because of presence of a chemical called pectin, right? Even though cellulose will be there, hemicellulose will be there, but because of pectin, it is tough. And I am sure you must be touching the bark of the tree with your palm, and you will feel how tough the cell wall will be, isn't it? And it is present only in plants. It is not present in us. There is no cell wall in animals, including human beings. That is one of the fundamental differences you have studied in junior classes, isn't it? So it's a non-living, rigid structure found outer to the plasma membrane, both in fungi and plants. Whereas in animals, outermost membrane is plasma membrane. So cell wall has got a different chemical composition. In algae, it is made up of cellulose, galactans, mannans, minerals like calcium carbonate. Whereas in day-to-day plants that we see, it consists of cellulose, hemicellulose, pectin. See, just now I said. This gives toughness to cell wall, and of course many proteins. So cell wall of young plant cell, that is primary wall, is capable of growth. But as it becomes matured, it diminishes the growth and becomes a tough secondary wall, okay? And it's towards the membrane. When you take the section of that, how does it look like? Look at that. The structure of the cell wall will be like this. So basically, when you see the cell structure as such, you can see the green colored one, cell wall labeled. I told you it is like a swimming pool, Science School swimming pool, cytoplasm inside, and all cell organelles are interconnected. You have to just imagine, through all the tubes, okay, car tubes inside your swimming pool. From top, you go to first floor of your gymnasium and see, look at the swimming pool, how it looks like. Similarly, here cytoplasm is the liquid, and in that all those tube-like ones are the cell organelles, okay? And look at that, when you take the section of it, you got a cell wall, right? 1 minute, I'll just unlock the room. Yeah, okay. So it is the plasma membrane, outermost one is the cell wall, then plasma membrane. And when we take the cell wall, the structure of cell wall will be like this. Look at that. There is a plasma membrane at the bottom, there is a primary cell wall and middle lamellae, finger-like structures. And you can see the presence of pectin. I said, what makes cell wall tough? It is the pectin. And then there are crosses, you see. Those crosses are glycon, again another chemical. And then of course cellulose is very much present in the form of small thread-like structures. They are called microfibrils. Okay, and then you can see the same structure. Okay, so it is the pectin that makes it tough, and the middle lamellae. Okay, right. So the middle lamellae is a layer mainly of calcium pectate. What is the function of this? It makes the neighboring cells adhere together. Glues means joining together. The cell wall and middle lamellae may be traversed by small, finger-like structures I have shown you just now, plasmodesmata, and this connects cytoplasm of the neighboring cell. Cell to cell will be joined by plasmodesmata. What is the function of the cell wall? As you guess, as you have been seeing it, it gives shape to the cell, and main thing is it gives protection to the cell from mechanical damage and infection. It helps a cell to cell interaction. One cell will interact with another cell through cell wall. It acts as a barrier to undesirable macromolecules. If some big molecules, macro means big sized molecules, want to enter inside, this becomes like a obstruction. Just like in Sunday school gate I said, the guards are there, no? So these guards will not allow unauthorized persons inside. Like that, cell wall will not allow the undesirable big sized molecules. And we have certain cell organelles wherein they are interconnected. Okay, one cell organelle is interconnected with the other, like endoplasmic reticulum. Okay, ER we abbreviate it. In my previous session I said the surface of the endoplasmic reticulum, if it is made up of, or if it provides surface, its surface to ribosomes, then we say rough endoplasmic reticulum. Look at that, I am showing here. I hope you are able to see. RER. And that small dots are there, you see, on the surface. Those small dots are ribosomes. So if ribosomes are present on the surface of the ER, then the ER becomes RER, rough endoplasmic reticulum, as you are seeing. Whereas if the surface of the endoplasmic reticulum doesn't provide its surface to ribosomes, the surface becomes smooth. Hence, the ER is called SER, smooth endoplasmic reticulum. And basically, as you are seeing, endoplasmic reticulum forms a bridge between nuclear membrane and cell membrane. Look at that. The nuclear membrane and cell membrane are being attached by this endoplasmic reticulum. And you can see there is a network of tiny tubular structures scattered in the cytoplasm. ER divides into two compartments. One is the inside ER, another is the cytoplasm. We say luminal and extraluminal. So there are two types, as I said, rough and smooth. So rough endoplasmic reticulum, it has got ribosomes on the surface. That's why the surface becomes rough and we say rough endoplasmic reticulum. And what is the main function here? You must note down here. The main function of RER is protein synthesis. Okay? Protein synthesis is the main function of RER. Why? Any guess? Any guess, Naveen? Rajwal?
Ribosome.
Yeah, right. Very good. Ribosomes are present on the surface of it. That's why protein synthesis. And it is also responsible for secretion of certain enzymes. that help in various activities. Whereas the smooth one, SCR, it's also important. It doesn't have ribosomes, so there is no question of protein synthesis, but it synthesizes lipids. Lipids means fats. So fats are synthesized by SCR. So you must remember this difference between RER and SCR. RER surface will be, ribosomes will be present on the surface of RER. On the surface of SCR, no ribosomes will be present. Hence it is SCR. RER is responsible for protein synthesis and secretion of certain enzymes. SCR is responsible for lipid synthesis. Okay, and you can see some hormones also are synthesized in SCR. So you can see the difference between RER and SCR, and it is present across the cytoplasm. I hope it's clear. Now we go to another membranous one. This is called Golgi apparatus. Now look at that Golgi apparatus, how it will be. It is made up of vesicles and cisternae. Cisternae means tube-like structures. So tubes and what you can bubbles, we can say. So Golgi apparatus basically is made up of two things: tube-like structures called cisternae and bubble-like structures called vesicles. Hope it's clear. And it is named as Golgi apparatus or Golgi body because of the name of the scientist who discovered it. So the gentleman's name is Camillo Golgi, way back 1898. Look at that. So it is densely strained reticular structures near the nucleus, and it is first observed by Camillo Golgi. It consists of, as I said, flat disc-shaped sacs. Those sacs are called cisternae. Okay, cisternae, and these are stacked parallel, as you are seeing. They are very small, 1 nanometer, and cisternae are concentrically arranged with the convex face. That's why we are saying cis face, and down we say trans face because it is concave shape and it is matured. So at the bottom, at the trans face, the cisternae are matured, whereas on top it is immatured. Cis face side it is immatured. Okay, is it clear? And the cisternae on maturity, as you are seeing, will form droplet or round structures, spherical structures. You can see here in the diagram. And on maturity, these round structures will get cut off from the tube-like structures and form vesicles. So basically, Golgi apparatus is formed out of two structures. So what is the function? It secretes materials to the intracellular targets. Intra means within. So whatever materials in the form of minerals are required within the cell, means inside the cell, intracell, this will supply. And it supplies to outside also. That means it is mainly transportation. So materials to be packaged as vesicles. Vesicles are round structures. From the ER, fuse with the cis phase and move towards trans phase. And this is why Golgi apparatus remains in close association with ER, endoplasmic reticulum, right? And the formation of glycoproteins and glycolipids means both proteins and fats are being synthesized by Golgi apparatus. And you can see those vesicles that are formed from cis phase to trans phase, and in between there is inside some empty space. That empty space is called lumen. Okay, and they are secretory. These round vesicles are secretory in nature. They secrete glycoproteins and glycolipids. Then we move on to the third one, it is called lysosomes, and most of the time this question will be asked in your UPSC NDA as well as your NEET exam. What are suicidal cells of the, or what are suicidal structures of a cell? So your answer should be lysosomes. Why suicidal? They collect all the waste material, go to the surface of the cell and then blast off. Since they are blasting off, they are breaking off, it is almost like committing suicide. Hence they are called suicidal organelles of the cells. Okay? And basically they are involved in throwing out waste materials. So they are also membrane bound vesicular structures formed by the process of packaging in the Golgi apparatus. Lysosomal vesicles contain almost all types of hydrolytic enzymes. Look at that. They have got enzymes. That's how they are able to digest, like hydrolases, lipases. Lipases, enzymes that dissolve or digest lipids. Proteases, enzymes that digest proteins. Okay? Carbohydrates or carbohydrases, enzymes that digest or break carbohydrates like sugars. So they are active at the acidic pH. Only condition is there. What is that? The digestive part should have pH acidic. You know, I think you must be, your chemistry teacher must have told you pH, negative log of hydrogen ion concentration, we say. And there is a scale 0 to 14. So 0 to 7 is acidic, and then 7 to 14 is basic or alkaline. Exactly 7, around 7 is neutral. Right? Here the lysosomes require acidic pH medium. Then only they will be able to be active and digest carbohydrates, proteins, lipids and nucleic acids. Is it clear? Next, similar structure, they are also round, just like lysosomes. These are called vacuoles. So there will be many spherical structures inside the plant cell as well as animal cell, and they are membrane bound. Since they are membrane bound and present inside the cell, we say endomembrane system. And what you must remember is that vacuoles is bound by a single membrane, and the name of that membrane many times he will be asking you, even in CBSE as well as your UPSC. Tonoplast. Remember? Tonoplast. The outermost membrane present in the vacuole is called tonoplast. And what does the vacuole contain? It contains water, sap means cytoplasm, excretory products and other materials not useful for the cell. So they are also like lysosomes, throw out waste materials. All wastes are thrown out. In plant cells, as we have seen, all of you have drawn the diagram also. Vacuoles will be very big in size, and being big in size, they push the nucleus to one side, one of the sides of the cell, and they are small in number, maybe 2, 3. Whereas in animal cell, vacuoles will be very small in size and many in numbers. So in plant cells, the vacuoles can occupy up to 90% of the volume of the cell. Look at that. What is the fate of other organelles? Only 10%. Right? That is the domination of vacuoles. So in plants, tonoplast, that is outermost membrane of the vacuole, facilitates the transport of ions and other materials against concentration gradient into vacuole. Now, what do you mean by concentration gradient? In my previous session, in my previous class, I said, talking about diffusion, talking about osmosis, I said, one side number of molecules are more, other side number of molecules are less. We said the molecules which are more, that area we said high concentrated area. Molecules which are low, we said dilute area or low concentrated area. So the difference between the two concentration is the concentration gradient. Okay. So this tonoplast will make the materials to move against the concentration gradient. That is the speciality of this. That means normally molecules from higher area move to low area without spending energy. Passive, we said passive transport. But here, if the molecules have to move from low concentrated to high concentrated, they have to spend the energy. If energy is spent and molecules are pushed from one area to another area, then we say active transport. So here the tonoplast is doing that, right? So in amoeba, the contractile vacuole helps in excretion. In many cells, food vacuoles, many of the protists, remember particularly the paramecium, the vacuoles will absorb the food will be in the form of an insect, and the vacuoles will keep on rotating. We said cyclosis. As the food vacuoles rotate, the insect gets digested or the food gets digested. So since vacuoles are helping the paramecium and other organisms in digesting the food by engulfing the food and rotating along with the flow of the cytoplasm, we say food vacuoles. So vacuoles can be excretory vacuoles as well as food vacuoles. Okay. Now we come to the most important one that we have been talking, mitochondria. And what we said, mitochondria are the powerhouses of the cell. Again, this is number of times asked. What are powerhouses of the cell? Mitochondria. Mitochondria plural, mitochondrion singular. And why are they so called powerhouses of the cell? Because they store energy in the form of ATP. I said ATP is the chemical form of energy. And the expansion of ATP, adenosine triphosphate. Okay, adenosine triphosphate. So lot of ATP molecules will be stored in mitochondria. Hence, it is called powerhouse of the cell. So we must know the anatomy of it. To know the anatomy, what is inside, we have to take a section of mitochondria and observe under electron microscope, not our compound microscope. Then we can see the structure like this. See, basically there is outer membrane, smooth outer membrane. And if you see the shape also, it is something like oval shape or disc shape, overall. And look at the inner membrane. If you observe carefully the inner membrane, it is forming finger-like structures. They are called cristae, crista or cristae. And inside the mitochondria, there is fluid called matrix. Right? The inner area of mitochondria is called matrix. So in the matrix, the inner membrane is forming finger-like structures. Those finger-like structures are called cristae. And in between the cristae, there is some space. It's called inner membrane space. So where exactly this ATP will be stored? In the cristae. Again, of course, when I go to higher classes, in the cristae there will be F0, F1 particles. So those F0, F1 particles will store the ATP. Now since it is storing the ATP, we call this as powerhouse of the cell. In the second diagram you can see further, you can see, yes, he is showing including the F0, F1 complex, which I talked just now. So outer membrane, first to focus on the outer membrane. I hope you are able to see. Then focus on the inner membrane and look at that finger-like structures, infoldings of inner membrane. That infolding membranes will form finger-like structures. They are called cristae. And the whole inner space is called matrix. So in the matrix, you can see the formation of cristae. And there is presence of DNA and ribosomes. You see, This is the specialty of mitochondria. Cell will have DNA because cell will be having nucleus. Inside the nucleus, DNA, RNA will be there. In spite of cell having DNA, mitochondria will have its own DNA. That is the specialty of mitochondria. Okay? So you must remember, sometimes he will ask you a question. Along with the cell, which other organelle will have its own DNA? You have to name it as mitochondria. And as I said, that finger-like structures called cristae will have small bulb-like structures. Let me expand this. You are able to see that? The green ones. Okay? Small pebbles like. These are F0, F1 particles. And many particles are accumulated in one place, then we say complex. F0, F1 complex. This is the site of storage of ATP. That is why we call it as powerhouse of the cell. Now, mitochondrion is singular, I said. It's a double membrane bound structure with outer membrane and inner membrane. It divides lumen into two aqueous compartments, outer compartment, inner compartment. That inner compartment is called matrix. And inner membrane forms number of infoldings. I said finger-like structures. They are called cristae. Remember Krishna, Lord Krishna? So cristae towards the matrix. They increase the surface area. So what is the purpose of this infolding of the inner membrane or formation of cristae? This is to increase the surface area. So the two membranes have their own special enzymes associated with mitochondrial function. So matrix possesses a circular DNA, a few RNA molecules and ribosomes, as we have seen just now. And these are mainly responsible for protein synthesis. So mitochondria gets divided by fission. Already you know fission means the parental body breaks off and forms two daughter cells. Each daughter cell will become an adult one, like that. So what is the function of mitochondria? Just now we discussed. It is basically a site for aerobic respiration. And at the end of aerobic respiration, what is aerobic respiration? Oxidation of glucose. See, Naveen, whatever we eat in the form of breakfast, lunch, dinner, snacks, remember we said BLD. B for breakfast, L for lunch, D for dinner, etc., etc. You are eating snacks, no? BLDE. So at the end of this BLDE, what happens? You get glucose. You see, end product of digestion is always glucose. And when this glucose gets oxidized, what do you mean by oxidized? Glucose combines with oxygen. When it combines with oxygen, the glucose gets broken. On breaking of glucose, you get energy. That energy will be in the form of ATP. And to be specific, one glucose molecule on oxidation forms 36 ATP molecules. And of course, your physics teacher will be telling you one adenosine triphosphate, when we break, you get something like 4 kilocalories of energy or 4 calories of energy. So when we eat so many breakfast, lunch, dinner, etc., etc., imagine how many lakhs of glucose molecules. And that many lakhs of glucose molecules into 36 ATPs, into 4 calories. See, this much energy is produced in our body. And what is important? The energy that is formed should be utilized. Then only you will maintain the body. If you are not able to utilize, what happens? It gets stored. And particularly it gets stored in the form of fats. You become fatty. Now since mitochondria is capable of storing that energy, ATP, they are called powerhouses of the cell, KEB. Karnataka Electricity Board, like that. Now once again you can see that cistrae and cristae and F0, F1 particles. You can see the matrix, inner space, presence of DNA, that is the unique property of mitochondria, and ribosomes. Clear? Now we go to the plastids, another important cell organelle. Basically, plastids are storage organs. Just like for mitochondria, we said mitochondria are the powerhouse of the cell. Plastids are the kitchen of the cell. Adike mane. Your mommy prepares food in the kitchen, no? And from the kitchen, the material comes to dining table. You start eating. Here also, for all the plant cells, it is the plastids. And these plastids are present both in animals as well as plants. But only thing is depending upon the variety. What is that variety? They are basically of three types. As you are seeing, chloroplast, chromoplast, leucoplast. Okay? So look at that. Chloroplast, you are able to see green color. Why green color? Any idea? Prajwal? Why chloroplast? Yeah. Yes, there is a green color pigment called chlorophyll will be present in chloroplast. Hence, chloroplast will be green in color. Since all the parts in the plant, except perhaps stem, all the plants and root, of course, predominantly leaves, are green in color. Whatever green portions you are seeing in the plant, you must remember, they are green because of presence of chloroplast. And chloroplast will have chlorophyll. That's why plants are green. And chromoplast, you must be seeing in your own home, in your garden. Colorful leaves, a plant having, say, top yellow color, middle brown color, down green color, like that. These are chromoplast, means coloring one, other than the green, chromoplast. And look at the leucoplasts. Leucoplasts are colorless. They don't have color. What do? They simply store. They store starch, leucoplasts. What about in our case, Naveen? Do we have all this? Chromoplast, chloroplast, leucoplast? Raghavendra?
No, sir,
not there, no? Had we got it, how nice, no? Had we got all of us chloroplast, we would have been appearing green, all of us.
would have been
Photosynthesis. Yeah, we will be doing our own food, but only thing is, Naveen will have problem. What is that? Breakfast starch, lunch starch, dinner starch, snacks starch, everything starch. Could you enjoy? No? I am sure you are all enjoying your mummy's food. She is preparing one day puri, another day idli, another day dosa, another day pad, uppettu. No, you are all enjoying varieties at home. So thank God that we don't have chloroplasts. Otherwise life would have been boring. So imagine plants. That's why we must show pity. They don't have variety. Every day starch. The day they have taken birth until death, chloroplast, chloroplast, chloroplast. Starch. Okay? Right. So basically, yeah?
It just leaves, sir.
Correct. From there, transportation takes place now. Starch prepared in the leaf will be transported by phloem. Remember xylem phloem?
We have so many things to do in life, sir. Only live now, sir.
But plants are there.
Same variety of food is enough for them.
But because of plants, we are surviving. Don't forget that. Just imagine the world without a single plant. What would be our life? Where is oxygen? Come on. We are able to breathe oxygen thanks to the plants. So be thankful to plants, and as far as possible, put at least 10 plants in and around your house. Then you have nice oxygen. Forget about the starch. But remember, today after my class, you just go to kitchen, see your mummy, what she is doing, how many vegetables she is cutting for your breakfast and lunch, dinner. All are plant products. Don't forget that. Isn't it? What about fruits that you are eating? Hence, we must be thankful to plants. Right. Tomorrow we will discuss about these plastids further. Right. I think it's time. We'll see you tomorrow.